So today we'll be talking about Mendelian genetics. But before that, I just have a couple of announcements. When you guys email me, definitely put in the section time. Like, if you guys say your section on Wednesday, I have 3 sections on Wednesdays. So include the time. In your case, it will be 7:30 p.m. But yeah, I have 74 of you guys, and it makes it a lot easier to figure out everything for you guys if I know which section you guys are in. Okay. So quiz 1 and 2 will be closing tonight. If you guys haven't done so already, please do it. I've had some students email me regarding these 2 quizzes that they're not apparently showing up on Blackboard. Can you guys still see it or no?
I usually do it the day after class, so I'm not sure. I haven't checked since I did it.
Okay. It's very odd because I have it available for everyone, but it's not showing up with some students.
I mean, it shows quiz number 1 and 2 in both modules for me. I just checked it.
Okay, at least you guys can see it. Okay. Okay. So quiz number 3 will be up after the session once I get everything sorted out with these videos. Again, I keep my quizzes conceptual, so don't freak out after the session, okay? Okay. So remember that question that we were talking about with the cell cycle? I actually got them mixed up with another question as well. So during which stage can cells exit the cell cycle or become committed to completing the cell cycle? So as you guys said, it is actually G1, where you go out to G0. S phase is where you end up getting irreversibly committed to the cell cycle itself, okay? Okay. Okay, so as we had a question regarding the supercoiling and linking number and all that fun stuff, I added 2 questions on here just to give you guys an example. So a circular bacterial DNA that is 2,000 base pairs in size has a linking number of 100. What does this tell you about the DNA molecule itself? Assume a Watson and Crick DNA structure of 10 base pairs per turn. So looking at this, what do we know? What can we actually know about this molecule? Any ideas?
It turns 10 times.
Oh, somebody was saying something and then got muted again. What happened? New?
I was saying like you have the complete 10 of 10 times.
Yes, so essentially with, so linking number. Linking number is the number of right-handed turns, 1 DNA strand. So in the double helix, you have 2 strands, right? Linking number is actually the number of right-handed turns that one of those strands do around the other, okay? So you have 100 right-handed turns on that specific DNA strand, okay? Then you have twist. Twist is equal to the number of base pairs over the number of base pairs per turn. So in this case, you have 2,000 base pairs, and you divided that by 10 base pairs per turn, right? So that gives you 200 turns. Does that make sense so far?
Oh, wait. I have a question.
Yes.
Can you hear me?
Yes.
I can hear you.
Okay, if you want, you can put it in the chat as well, the question.
Do you always divide by the 10 base pairs per turn?
So it depends on the question itself. If it says that assume a Watson and Crick DNA structure of 10 base pairs per turn, then you would divide it by 10. The question itself will tell you how many base pairs per turn there is, okay?
Okay, thank you.
Yep. You okay, Rafiq? Okay, so the next part to this question is ripe. Oh. So ripe is an expression that goes with the number of supercoils, okay? So that's where this equation comes in. So this is the same way you can determine any of the three. With this question, ripe is equal to the linking number minus the twist number, okay? So that's how you would end up with right equal 100 minus the number of twists, which is 200, which gives you negative 100. So a right of negative 100 just means that you have 100 negative supercoils. So if this number ended up positive, it would have been 100 positive supercoils. And if it ends up 0, then there is no supercoiling going on. It's just there. Does that make sense?
What's the difference between a negative and a positive supercoil?
So negative supercoiling is very underwound. So you know how coiling is just like okay, so I'm gonna use my charger here. Can you guys see my charger?
Yeah.
So supercoiling is just a situation where it's like going like this, right? All wound up, right? So when it's positive supercoiling, it's overwound to a point where it's like like this. Does that make sense?
Yeah.
Whereas with negative supercoiling, it is so underwound that it's like kind of going that way. At 0 supercoiling, it's just relaxed but not underwound or overwound. Does that make sense?
Yeah.
Okay, great. Okay. So this is another question, except with 0 base pairs, not 0, 1,000 base pairs. Can't read today, apparently. It's been a long day. Anyways, so again, 100 for the linking number. This is the way you would do that. it gives you 100 turns per twist. And then the right, you end up with 0 because 100 minus 100 is 0, which means it is not supercoiled, okay? It's not positive or negative. It's just at a stable state, just sitting there. Does that make sense?
Yes.
No.
Where am I losing you?
Honestly, everything.
Okay,
Sorry.
oh. Wait, what were you saying?
Sorry.
Oh no, don't be sorry. So linking number. So essentially these 3 terms come into the description of what a DNA molecule is up to, right? So essentially it is trying to see how it's twisted, how it's kind of wrapping around each other, right?
Yeah.
So linking number, we have it as the number of right-handed turns. So it's so we have a DNA strand going right-handed turns, right? Other than Z DNA, but that's another story over there. But so for now, just like linking number is the number of right-handed turns of one DNA strand around its complementary strand.
So is it always 100, or is that number always 100?
No, it can change depending on the question. This is just one. This is just an example that I just randomly took out. It can be any number. It's just, this is just a relationship. So the relationship for a linking number is twist plus right. So does that make sense?
Yeah, thank you.
So does that kind of give the picture of what's going on?
Hello?
Yeah.
Yes.
Can you hear me? I think I was muted. Yeah.
Okay, good.
Thank you.
Any other questions about this guy?
No.
Okay,
Nope.
good. I'm happy. Okay, so now we're gonna go into presentations. I have about 4 of you guys today, so whoever wants to go first can go.
It's saying host disabled.
Oh, whoops, my bad. I really got to do that before. And anyways, can you, yep, perfect. Okay. Ooh, pretty.
Thank you.
I like the font.
Yay. So I got the question. Hi, my name is Rama. I'm gonna talk about DNA structure and organization. Basically on Avery, McLeod, and McCarthy experiment. So basically what the experiment talks about is they took the virulent cells and then refined it in a liquid medium with large quantities of cells, and then they collected it and then killed it with heat. And then they took some of these the filtrate and then put the avirulent cells, some pools of the avirulent cells into the filtrate, and then they divided it into 3. One named the protein, right RNA and DNA. So they treated these filtrates, the first one with protease to see if the transformant molecule was protein, and then there was no reaction, that means that was not the transforming factor. And they did the ribonuclease in RNA and realized that was not a transforming factor. And then they did it for DNA, and then there was they realized that that was the transforming factor. So they concluded that DNA was a transforming factor, and it was responsible for transforming genes from offsprings to offsprings. So the question I got was, if Avery, McLeod, and McCarthy had determined that the transforming molecule was a protein, what experimental results would have been observed? My answer is highlighted in red. Say, he killed cultures treated with protease would not have transformed the R cells. Does anyone have questions? No? Okay. Thank you.
Good. Are we good with this question, guys?
Mm-hmm.
Okay, good.
Yeah.
Who wants to go next?
I guess I'll go.
Okay.
All right, so let me see how to screen share here. All right. Is it doing it yet? Oh, all right, good. Oh. Can you see it?
Yes.
All right, perfect. All right, so I had question number 1. My name is Brandon, and pretty much my question was, how many different kinds of gametes can be produced by an individual with the genotype well, A, B, B, C, C, D, D, E, and F, F. Obviously, different ones are capitalized. And so in this situation, 2n, 2 to the nth power, is equal to the number of gametes an organism can produce. The n would represent the number of different chromosomes / heterozygotes present. And the capital letters represent the first homologue, and the lowercase letters represent the second homologue. And in this specific case of the problem, we have the genotype listed there. We have 3 different chromosomes in this genotype: the capital B and lowercase b, the capital E, lowercase c, and capital F and lowercase f. So our equation to find the different kinds of gametes produced would be 2 to the 3rd power, which would equal 8. In the end, the answer would be 8 different kinds of gametes, and the answer is right there. Does anybody have any questions?
It's always heterozygotes, right?
What did you say?
Like you only count the heterozygotes, so you wouldn't
Yeah,
count
you
the
only-
capital
you
or
only-
capital.
Yeah, you only count the heterozygotes. So like over here, you wouldn't count like the ones that have only capitalized letters or only lowercase. It has to be like a capital letter matched with a lowercase letter, and that's how you do it.
Okay, thank you.
Of course, yeah.
So with this specific type of question, since it's only showing the genotypes, that's when you go with the heterozygotes. If you
Yes,
have
yeah.
like the chromosome number, then it would be the chromosome number that you would actually use.
Mm-hmm.
Yep.
Alrighty.
Good work.
Thank you.
Who's next?
You tell me the
I'll go next. Can you guys hear me?
Yep.
Okay. So can you see the slides?
Yes.
Okay, sorry, I was on the wrong one. So mine was the 3rd question for today.
Okay.
So a brief little beforehand. What is synapsis? It's an event that occurs during prophase 1 of meiosis, in which homologous chromosomes are paired up, and after that, crossing over can occur. So my question was, what is the outcome of synapsis? And the answer is right here, which is side-by-side alignment of homologous chromosomes.
Good.
All right. Any questions, anyone?
Nope.
All right. Awesome.
Gabriella? Gabriella? Okay, so I'm not sure what's going on. Yes, please. Definitely have your videos on, guys. Okay.
I don't have a webcam.
That's okay then.
Okay.
Gabriella, I have you listed down for presentations today. Are you still going? Did you disappear on me? Okay. I guess we'll continue with what we have planned for discussion. If I can't find you. Okay. 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 Can everyone see my screen again? Okay.
Yes.
Oh. Okay. So now we're going to get into some Mendelian genetics. So what are the postulates in Mendelian inheritance, or the laws of them?
It's based off of laws of probability.
Okay, so that definitely does happen. But the laws themselves are specifically these guys. So we have the law of segregation, which is dependent, which is just saying that alleles of the same gene segregate from each other. They separate out each other. Then you have law of independent assortment, which means that genes for different traits can separate out independently. And then you have the law of dominance, which means that some alleles are dominant while others are recessive. And in this case, if you have an organism with at least one dominant allele, it displays the effect of the dominant allele. Okay. And then obviously we know that alleles exist in pairs because as we know, we have homologous chromosomes, one from the mom, one from the dad. So in that case, you end up with 2 alleles each gene, okay? Great. Okay. Technology does not like me today. Okay. So to figure out genotypic and phenotypic outcomes, the probability of those guys, you have two different rules going on. The first one is the product rule. It states that the probability of independent events occurring together is the product of their individual probabilities. So for an example, if you're rolling 2 dice, the outcome of one die is independent of the other, okay? So if the probability of rolling a 4 is 1/6 because you have 6 sides on a dice. And
I have a
overall,
question.
oh, yes.
If you're
Okay,
is it working now?
yes.
Okay. So if you're rolling like 2 dice together, right? And you roll them like together, all together, like not separately, they could hit each other, and then the outcome will be affected by both distance. So are they like individually different? Don't they
Yes.
affect each other if you throw them together?
Even if you're throwing it together, the outcome will be independent of each other because for one thing, it's 2 different dies, and it always comes out to be 1 out of 6 for each of them because it's 6 sides with 1 possibility of getting a 4, right?
Okay, but okay.
So probability of rolling 2 4s would be 1/6 times 1/6. So it's kind of like so it's 2 separate events, and the one choice is not influencing the other. Next we have the sum rule. The sum rule states that the probability of either of 2 mutually exclusive events occurring is the sum of their individual probabilities. So it's kind of like an either or situation in this case. So the probability of rolling either 2 4s or 2 3s will end up this plus the probability of 2 3s, which gives you 1/18. Does that make sense? So it's like an either or situation where order doesn't really matter. Wait, order does actually matter in this case. Does that make sense?
I'm kind of confused with the sum rule. Can you go over that again?
Okay, so the sum rule, it's kind of like, so when we do this, it's either of 2, so it's kind of looking at the probability of either of 2 mutually exclusive events. Okay, so I feel like this would make more sense so in this case, you're kind of going with, you're trying to figure out the probability for 2 4s, like getting 2 4s or 2 3s, right? So the probability of getting 2 4s is 1/36, as we know. And then we have the probability of getting 2 3s is 1/36 again. So to figure out kind of an either or situation, you would add those together and you would get that answer. Does that make sense?
Oh, yes. Thank you.
So another example that looks more genetics than statistics is this question here. In a family with 2 children, what is the probability of there being 2 females? So what do you guys think? We have
Wouldn't
a little
that be
wait, say that
1/2 times 1/2?
So that would
Would
give us 1/4, right? How about 2 females or 2 males? Which rule would come into play?
Would you just do 1/4 times 1/4?
Close,
Plus
but
one.
we, oh. Chat coming up. Yep, it is the sum rule. Yep. So yes, it becomes half. Does that make sense? It's like or sum rule. Okay. What about this guy? 1 female and 1 male. What do you guys think?
I think that's also going to be 1/4.
Okay, so in this case, since it's, let me just check the chat. Yes, okay, it will actually be 1/2 actually, because in this case, it can be any order. So you gotta account for any of the orders, all of the orders. So in this case, you would only have 2 different orders. So that would give you each of these guys would get 1/4 of a probability, and then you end up adding them together to get the total, right? So that would be your probability for that. Okay. So what do you guys know about monohybrid crosses?
Not much.
Wait, what's happening? Did somebody say something?
I said not much.
Okay. Let's get you to something in them. Okay, so monohybrid crosses. They are essentially a mating between individuals who have different alleles at a certain genetic or gene, okay? So in this case, these 2 guys have heterozygous genotypes, right? So like, okay, let's cross these guys. What do we get? This is a Punnett square. It will come in handy when you guys do questions like this. So to figure out which one comes out where, so you have both of these genotypes, one from the male one from the female one. You get 4 different types of groups, right? Well, not 4 different types, but you're looking at 4 different, 4 progenies, I guess. And it's based on 1 gene or trait or genetic locus of interest, whatever you want to call it. So the probability of getting a homozygous dominant is 1/2 times 1/2, because you have 1/2 a chance of getting a big P from the male and 1 big P from the female. Same goes with the recessive guys. You get 1 from each parent. So that gives you 1/4 as well. Then you get this value where with the heterozygous, again, remember order matters. So there will be combinations that can come out. So 1/4 and 1/4 gives you a 1/2. So that is the genotypic ratio for this number. So 1 to 2 to 1 is the genotypic ratio, and you get a 3 to 1 phenotypic ratio, okay? Now, what do we know about dihybrid crosses? Thinking about what we just said with monohybrid.
Instead of 1 allele, it will be 2 alleles. Then.
Close, not alleles, but genes or traits, right? Because in monohybrid, we're looking at 1 trait. In dihybrid, you look at 2 traits. And in this cross, since we're looking at two different traits, that's how Mendel came up with the law of independent assortment, essentially saying that the inheritance of one locus has no influence over another. Okay? And this shows a 9:3:3:1 ratio. However, it can be modified depending on the conditions it has, but we won't go into the factors that are involved just yet, okay? So don't rack your head on that too much. Just remember this phenotypic ratio that you see, and then the law of independent assortment, okay? That's how it gets shown. So this is a question that involves these crosses. Mendel crossed peas having round yellow seeds with peas having wrinkled green seeds. All F1 plants had seeds that were round and yellow. Predict the results of test crossing these F1 plants. So, oh, wait, okay. So what do we know from this question? Yeah, okay. So, okay. Okay, hold on, sorry. I'm having a little bit of technical issues today. Okay. Okay. So we have these, so again, how you define it depends on how it goes, okay? So since you know that all of them ended up round and yellow, you know those traits are actually the dominant ones, right? Yes, that is correct, Trace. Okay. Oh, wait, did I miss something? Okay, yes. So yes, those are the dominant guys there. So we know that so that exists, and we have a heterozygous situation. But if we didn't know that, we would be like, okay, so how would we actually figure out the zygosity for this? We do that by a method called test cross. So test crossing is a test in where you breed a phenotypically dominant individual with a phenotypically recessive individual to actually determine the genotype of that phenotypically dominant individual. So answering this question ends up with looking at the different, so you take this one and you see that with the homozygous recessive individual, and then you see that you would get half round, half wrinkled. In this case, you would see that if you had a Y, big Y, little y, that would be half green, half yellow, okay? So in this case, when you look at the probabilities of the different phenotypes, you would go with, okay, so round has half, yellow has half. You multiply those together and you get 1/4. And then you continue down with all of those, round and green, half and half, that equals 1/4. So just kind of going through that. I feel like the next question will actually give you more of an idea as to how test crosses work. So if you had a fruit fly that was phenotype dominant A, would you, what test would you make to determine if the fly's genotype was homozygous dominant or heterozygous? So we would use a test cross, right? Because we're trying, oh. Yes, oh. So we would test cross it or cross it to the recessive allele, right? The recessive phenotype, Drosophila. So if the fly had homozygous dominant, all of them will be phenotypically dominant with heterozygous progeny. So all of them would be dominant. If it was heterozygous, half of the progeny would end up dominant because, again, guys, just remember law of dominance. If you have one dominant allele, you end up with a dominant phenotype. And then you'll have half recessive individuals. Does that make sense? Okay. Which type of phenotypic ratios are likely to occur in crosses when dealing with two gene pairs for which all the genotypic combinations are of equal viability? What do you guys think? So what is the key point here in this question?
You're asking about phenotypes and genotypes. So the ratio for each of them, I guess.
Okay, so of what?
Oh, so a dihybrid. So wouldn't it be the 9331?
Yes. So make sure you guys read the questions carefully. So since it says two gene pairs, think dihybrid. If this said one gene pair, one gene, it would just be 1 to 2 to 1 or 3 to 1. That would be the phenotypic side, okay? For monohybrid. Does that make sense? Okay. Okay, so with human blood types, you have different alleles going on, right? So A and B are dominant. O is recessive, okay? So A has the homozygous and heterozygous version. Same goes with B. And AB, they're both dominant, so they both show up together. And O is recessive, so this is how you would note it with genotypes, okay? So this is like a donut version of blood groups, I guess. Just a way to visualize it. So type A only has A in it. Type B has B in it. AB has both. And then type O shows nothing, no antigens whatsoever, okay? So if you have a child that has the AB, blood group and the mother has AB, what are the possible blood types for the father? Have a response in the chat.
Wouldn't one of them be AB?
Yes. We also do get A or B, heterozygous or homozygous. I just put it like this for now, but at the end of the day, it's the phenotypes that they're asking in this question. So A, B, or AB. Does that make sense? Which ones get excluded from being the father?
Oh.
Yep. So if you have a father that's O and the child is AB, be like, no, no, he ain't the father.
Uh-oh.
Yep. Okay. So again, this is a different wording to the same exact question. Just a different way to write it. So again, the answer to this guy is these three phenotypically. It's like when, unless they ask like genotypes, then there would be more answers to this, but does this make sense? Okay. Okay. When I go over this, don't freak out on me, okay? I promise I'll even have videos for this. So, okay. So the binomial theorem is another way to calculate the probability of alternative ways to achieve a combination of events. So in this case, it involves 2, right? By 2. So it comes out, so this is the expression for the binomial theorem. I know you guys haven't covered this in lecture yet, so I'll have another video. Dr. Osuna will also go over it, but I wanted to make sure I got a question in here to make sure you guys understand. So A plus B, so A and B are the probabilities of the 2 alternative outcomes that you are testing, right? And N is the number of trials. So this is kind of the way of trying to cut down the number of times you do the product rule and sum rule altogether, just because it's so many different combinations when you get higher and higher levels of the number that you're looking for. So, okay, so before, obviously you guys can, okay, so don't freak out when you see this, but this is just an, I can already see people freaking out already. So don't worry, you guys will have this in front of you when you guys can actually do this, but just, ooh. Okay, good. So this guy, so if you go from 3 to, yes. Has everyone actually taken Calc 1?
I'm in it now.
Okay.
Same here.
Okay, good. So do you guys need me to go over how you would expand this guy out?
Yes.
Okay.
Yes.
Okay, so if you, so the number, the exponent that you use here, so the first term you would put it at a to the third, right? So if this is your third, you kind of go with the first number with your exponent. And then each term following, you decrease one for the first term and then add one to the second term without looking at any of the coefficients, okay? Then again, you don't, you kind of go up, down with the first term in exponents, up with the second term, right? Does that make sense? Okay. Now the coefficients, you don't have to memorize them. I'm also putting up a way that you can look at this without even remembering all of this, but you guys, for the exam, you can have this in front of you and be fine. Just remember it's always kind of like this, but yeah, you can have this in front of you. But I'll put up a video that also goes over how to get these coefficients as well. But for now, we're just gonna stick with whatever we have here, okay? Does that make sense so far? We'll go through an example so it's kind of gets a little bit less crazy. Okay. So what is the probability of a family in a family of 5 children that 2 will be male and 3 will be female? So in this case, I put down A equals the probability of male equal half, B equals the probability of female equal one half. Again, you can put it in any order as long as you be consistent with your exponents, because that whichever probability you use when you're plugging in, okay? And n equals 5 because 5 children would be the number of trials that you would be looking at. So in this case, you would take the a plus b to the 5th power and take that expanded form. And then when you take that expanded form, this, the number of males and number of females will be the exponents you will use. So the term you choose out of this guy will be based on the number of males and number of females that you end up wanting to look at. So in this case, since we have 2 males and I put down A as males, so you would take the one that has A square and B to the cube, okay? So the only option with that is this guy, 10A squared to B cubed, okay? So you plug in your probability values and then you get 10 over 32 as an answer. Does that make sense? Are we all on the same boat with this question? Okay, so there is one more thing I want to cover with you guys before you guys leave. So the chi-square test. I know you guys haven't covered this in class either. So the chi-square test is this formula here, where O is the observed value and E is the expected value. So it's the difference squared over the expected value, and you take the summation of all of those guys. So this is essentially a way to figure out the probability that this observed value, how much of a deviation this is from the expected values. So a question would be this guy. We have a sample size of 200 with the following genotypic ratios, not genotypic ratios, observed values. And we're trying to ask how much of a deviation that there is from the expected values, right? So we have these values. We're like, okay, we have the ratio of alleles, which we see that there is somewhat of a deviation from the 1-to-1 to 1-to-1 ratio, the expected ratio of it. So in this case, the chi-square test will end up allowing us to figure out how much of a deviation there is. In this scenario, there is, for the 200 progeny, you would use this ratio because you would divide this sample size by 4. So you would end up with 50 to 50 to 50 to 50, okay? So in this case, you would test a hypothesis that there is no significant difference between the observed values and the expected, okay? I know I'm going a little bit fast, but again, this is just calculating it out with this equation that we have. So kind of goes through each of them, and then you add these guys all up, because remember with our equation, it has the summation. So you're adding up all of these values and you get 9.88, okay? So now after this, you get this chi-square analysis table. And when you look at this guy, the first thing you need to do is figure out the degrees of freedom in the case. So in this case, you would go with the number of genotypic classes minus 1. So in this question, we had 4 genotypic classes, and you take away 1 from that, which gives you 3. So that's your degree of freedom here, right? So you come here and then you use that to figure out which is your p-value, okay? So we kind of look through the this row and see where it actually intersects. So with one of the things with this table is that you never go higher than the number, okay? So you never go higher than the number that you get as your chi-square value. So in this case, you would see that when you reach 2.5% or 0.025, you get 9.348, and at 0.01, you get 11.34. So you're like, okay, we are at 2.5%. That is our p-value. So the standard cutoff for these values is 5%, okay? So unless there is a question stating that it is not 5%, then it would end up with so this 5%, it shows that since our p-value 2.5 is less than the standard cutoff, the hypothesis that we made of no significant difference between our observed and expected values would be rejected. So it is saying that there is a significant difference between the values we have. Does that make sense? I wish. Genetics kind of takes the stats class into.
So are we gonna need to know all of this? How to get all this chi-square thing?
I think with what Dr. Osuna had in his concepts to know, you will definitely need to know it. Just like make sure you guys know the steps to actually do this question, because like he might ask you what is the chi-square value, or is how much of a difference. So it would be asking like the p-value for the question, you know. For my quiz specifically, I won't ask the map to this because I don't want to get you all freaked out. But definitely know how to do these, guys. I'll put up another video as well if you guys want me to, just to give you guys another perspective, I guess.
Yes, please.
Okay. I'll definitely find a video at some point before your exam next week. So any other questions? Oh, so since I threw a lot of stats at you guys today, I am going to let you guys, so in the chat, as long as you put down the word here, you guys get attendance credit. Still missing like 6 people. Guys disappeared. Missing like 3 more people. So any questions, concerns? Are you all ready to run away now?
Yeah.
Don't blame me, it is the class.
If I don't get it, should I switch majors?
I don't, I don't think it would come unless you specialize in like genetics itself.
No,
I don't.
I really wanted to leave stats in freshman year.
Yeah, I know. I know. It's a lot of information.
I have a question, but it's regarding one of the quizzes. It's an answer I got wrong. That was like talking about the chromosome. It gives like 2 numbers or something to identify where it is.
Oh, so hold on. Let me get the other PowerPoint for that. Okay.
Wait,
I think I did it backwards.
Is class still going or can we go?
Yeah, you guys can leave if you guys gotta go, but I'll get something. So.
Thank you.
Yep, not a problem.
She's laughing.
Okay.
My thing was.
Okay, so remember how we had so it goes chromosome number, the which arm it is, P or Q, and then band number. right? I think in your answer you had it backwards. You put band number, the chromosome arm, and then the chromosome number.
Where in the notes is this? I'm having trouble finding it.
It's in a video too.
It was
Yeah, you know, if you find the quiz password.
The
It was-
discussion video.
for quiz 2.
Was it quiz?
Yeah, for Kristen. Yeah.
Huh. Okay.
Does that make sense?
It will. I'm just gonna watch this. Thanks. Have a good one.
Okay. You too. Have a good night, guys.
You too.